continue with our part number 2 for the chapter number 9, that is strategies for enhancement in food production. And in part number 1 we will discuss the various aspects regarding this chapter, such as food is any nutritional substance that is required for the building and maintenance of the body, and food contains such as fats, carbohydrates and proteins. Animal breeding, a significant aspect of the animal husbandry, deals with the breeding of the animals, and further there are two types of the animal breeding, such as inbreeding and outbreeding. Inbreeding deals with the mating between the closely related members of the same species near about for 4 to 6 generations, whereas in outbreeding it deals with the mating between the unrelated species for about 5 to 6 generations. And next further important topic for this chapter is the animal husbandry, that is the common practices of breeding and livestock is known as the animal husbandry. And there are various aspects regarding the management of the farm and the farm animals, such as apiculture, beekeeping, pisciculture and farm animals. So these are the topics that are to be covered in the part number 2. And today we will start with our part number 2 with the plant breeding. So let us start with the plant breeding. Plant breeding is the practice of selecting and breeding specific desired plant species in order to obtain desirable traits such as more yield, pest resistance, herbicides resistance, etc. तो प्लांट ब्रीडिंग जो है वो एक ऐसा एस्पेक्ट है या एक ऐसी प्रैक्टिस है जिसमें कि हम क्या करते हैं, सिलेक्टिंग एंड ब्रीडिंग स्पेसिफिक डिजायर्ड प्लांट. किसी भी डिजायर्ड प्लांट की हम स्पेसिफिक जो उसकी ब्रीडिंग है उसको हम सिलेक्ट करते हैं इन ऑर्डर टू ऑब्टेन डिजायरेबल ट्रेट. और उसको सिलेक्ट करने का जो पर्पज होता है दैट इज टू ऑब्टेन द डिजायरेबल ट्रेट सच एज. अब डिजायरेबल ट्रेट्स में क्या क्या चीजें मिलेंगी, सच एज मोर यील्ड, pest resistance hoga, as well as herbicide resistance. So this is the most important criteria for selecting the desired plants for obtain the desirable traits in case of the plant breeding. The main steps in breeding, that is the new genetic crops are. अब जो main concepts हैं हमारे या main steps जो है breeding के, that is the new genetic crops are. First is collection of variability is the root of any breeding program. सबसे पहले हम क्या करेंगे, कलेक्शन करेंगे वेरिएबिलिटी की, इज द रूट ऑफ एनी ब्रीडिंग प्रोग्राम. इट इज एसेंशियल टू कलेक्ट एंड प्रिजर्व ऑल द डिफरेंट वाइल्ड वेराइटीज, स्पीशीज एंड रिलेटिव्स ऑफ द कल्टिवेटेड स्पीशीज फॉर इफेक्टिव एक्सप्लॉयटेशन ऑफ द नेचुरल जीन्स. तो इसके बाद ये एसेंशियल है, जरूरी है किस चीज के लिए, टू कलेक्ट एंड प्रिजर्व. सबसे पहले हम उनको कलेक्ट करेंगे. आफ्टर द कलेक्शन दे शुड बी प्रिजर्व ऑल द डिफरेंट वाइल्ड वेराइटीज, जितने भी वाइल्ड वेराइटीज होंगी उनको प्रिजर्व किया जाएगा. स्पीशीज को प्रिजर्व करना है एंड रिलेटिव्स ऑफ द कल्टिवेटेड स्पीशीज को प्रिजर्व करेंगे फॉर इफेक्टिव एक्सप्लॉयटेशन ऑफ द नेचुरल जीन्स. नेक्स्ट, अवेलेबल इन द पॉपुलेशंस, एंड दैट एक्सप्लॉयटेशन ऑफ द नेचुरल जीन्स होंगे वो किसमें प्रेजेंट होंगे, दैट आर अवेलेबल इन द पॉपुलेशंस. The entire collection, अब जो entire collection है, such as in case of the plants and the seeds, having all the diverse alleles, उसमें जितने भी होंगे वो सारे के सारे diverse alleles होंगे for all the genes in a given crop. और जितने भी diverse alleles होंगे for all the genes in a particular crop is called as the germplasm collection. Next, evaluation and selection of the parents. हम उसके बाद क्या करेंगे, इवेलुएट करेंगे. इवेलुएट करने के बाद सिलेक्शन करेंगे पेरेंट्स की विद द डिजायरेबल कैरेक्टरिस्टिक्स. अब उनकी इवेलुएशन जो होगी या सिलेक्शन जो होगी डिजायरेबल पेरेंट्स की, दैट इज ऑन द बिहाफ ऑफ द डिजायरेबल कैरेक्टरिस्टिक्स. सो नेक्स्ट इज द दिस इज द स्टेप्स that are involved in the plant breeding. What are the various steps that they have to be adopted for the selection of the desirable plant breeding? First is, this is the स्क्रीनिंग एंड सिलेक्शन. सबसे पहले स्क्रीनिंग एंड सिलेक्शन करेंगे, उसके बाद उसकी पॉपुलेशन डेवलपमेंट होगी, दैट इज द रिकॉम्बिनेशन एज वेल एज सेग्रीगेशन. नेक्स्ट इज स्क्रीनिंग एंड सिलेक्शन, उसके बाद हम उसकी स्क्रीनिंग और सिलेक्शन करेंगे. आफ्टर दैट देयर आर न्यू, आफ्टर द स्क्रीनिंग एंड सिलेक्शन देयर आर टू स्टेप्स आर टू बी फॉर्म्ड, दैट इज फर्स्ट इज द न्यू varieties आर टू बी फॉर्म्ड एंड सेकंड इज द germplasm resources आर टू बी फॉर्म्ड, such as cultivators, breeding lines, land races, wild relatives, etc. So these are the steps that are involved in the process of the plant breeding. Next is desired characters are now cross hybridized among the selected parents. अब उसके बाद जो desired characters होंगे वो desired characters होंगे they are cross hybridized. उन desired characters को जो है cross hybridized किया जाएगा among the selected parents. अब जो cross hybridized होंगे they are cross hybridized among the selected parents. This combine the characters genetically in the progeny. अब ये characters जो होंगे these characters generally combined in the process of progeny. This is a time consuming and tedious process. तो ये जो process है that is very time consuming as well as tedious process. Next, selection and testing of the superior recombinants. उसके बाद selection की जाएगी. Selection के बाद उनको जो है test किया जाएगा. किस चीज को test किया जाएगा of the superior recombinants. Those plants that contain desired characters are selected. जो plants desired characters को follow करेंगे उनको क्या किया जाएगा? उनको select किया जाएगा. Selection process is crucial to the success of the breeding objective and requires careful scientific evaluation of the progeny. उसके बाद जो selection process है, that selection process is very crucial to the success of the breeding. अब जो selection जो हमने की है process, that selection process is very crucial process in case of the success of the breeding object. किसी भी breeding objects के success के लिए जो है वो selection process जो है that is very crucial process and requires careful scientific evaluation. और उसके साथ साथ scientific evaluation जो है progeny की that should be must. Testing, release and commercialization of new cultivators. उसके बाद क्या होगा? Testing की जाएगी. Testing के बाद release किया जाएगा. And after the release, there is a commercialization of the new cultivators. The newly selected lines are evaluated for their yield. अब जो new selected lines हैं, they are used or evaluated for the yield and other agromatic traits of the quality, disease resistance, etc. And the testing of the new varieties and cultivars is conducted by growing them in the research fields. तो जो हमारी testing है new varieties की as well as cultivars is conducted by growing them in the research field. उनको हम कहाँ grow करेंगे? In the research fields. Their performance under ideal fertilizer applications. उनकी जो performance है, that performance is based under the fertilizer application, irrigation and other crop management practices. तो जो उनकी performance है that is under the ideal fertilizer, under the irrigation as well as other crop management practices is then recorded and analyzed. Next is plant breeding for disease resistance. What are the various aspects regarding the plant breeding for the disease resistance? Bacteria, fungi and virus affect different crop species. तो ये जो हमारे microorganisms हैं such as bacteria, fungi, virus, ये जो होते हैं वो different crop species को जो होते हैं वो affect करते हैं. Approximately 20 to 30% of the crops are lost. अगर हम average में देखते हैं तो near about approximately 20 to 30% crops होंगे they are lost due to the different pathogens and these crops are lost due to the different pathogens. Plant breeding is used to produce disease resistant plant. अब plant breeding जो है उसको हम use करते हैं या produce करते हैं for the purpose or to produce the disease resistant plants. To produce disease resistant plants, अगर disease resistant plants produce करना है, after that causative agent and mode of transmission of the disease should be understood. अगर हम किसी भी प्लांट ब्रीडिंग के अकॉर्डिंग हम चाहते हैं कि हम डिजीज रेजिस्टेंट प्लांट्स को प्रोड्यूस करें तो उसके पहले हमें क्या समझना पड़ेगा? दैट मीन्स पॉजिटिव एजेंट को समझना पड़ेगा. वी हैव टू बी आइडेंटिफाई द पॉजिटिव एजेंट एंड द मोड ऑफ द ट्रांसमिशन ऑफ द डिजीजेस. व्हाट आर द वेरियस मोड्स ऑफ द ट्रांसमिशन ऑफ द diseases that should be clearly understood. Next, methods of breeding for the disease resistance. What are the various methods for the breeding for the resistance of the diseases? Breeding can occur through conventional methods such as hybridization. So breeding जो है it can be occur normally by the conventional methods and that conventional methods such as hybridization and selection. So these are the two conventional methods that are used for the breeding, that are the hybridization and the selection. It includes steps such as, अभी कौन-कौन से steps को include करेगा, such as screening germplasm for resistant gene, hybridization of the selected parents, जो selected parents है उनकी hybridization को understood करना, selection and evaluation of the hybrids, selection के बाद उस hybrid की evaluation करना, testing and release of the new varieties after the testing, and after the testing there is the release of the new varieties. But the conventional methods has certain disadvantages. पर जो एक conventional method है, it has also certain disadvantages, such as in terms of the few disease resistant genes are known. जिसमें कि हमें few resistant genes जो कि अगर हम उनको known या उनको जानते हैं, तो that are the some disadvantages in case of this concept. Next is the mutational breeding. What do you mean by mutational breeding? It is one of the method of the plant breeding. Mutational method is also one of the most important method of the plant breeding that modifies base sequence, nucleotide sequence of the gene to obtain the new traits. जहाँ से कि वहाँ पर हमें जो है वो new traits होते हैं वो मिलते हैं other than those observed in the plants. These mutations or changes are introduced in plants. ये mutations और changes जो होते हैं, these are generally introduced in case of the plants either with the help of the chemicals or radiations such as X-rays. For example, mung bean variety is obtained through mutation, and this variety is resistant to yellow mosaic virus. So these are some mutational breedings that are related with the mutational breeding. Next is breeding to obtain insect or pest resistant variety. Breeding जो है that is to be obtained for the insects or the pest resistant variety. Insects or pest resistant plants are produced based on different strategies. ये जो है वो different strategies के ऊपर based करता है, such as they are modified in such a way that they become tasteless. इनको उस तरह से modified किया जाता है कि ultimately they become tasteless. So animals avoid eating these plants. जब वो tasteless होंगे on the behalf of tasteless, that means the animals they have to avoid to eat these plants. They produce some toxins that paralyzed the infected insects. ये कुछ toxins को produce करते हैं that directly paralyzed the infected insects. They are modified morphological such as hairer, pointed leaves, odor, etc. So plant breeding has helped to develop several useful and important varieties of the certain crop plants. तो इस वजह से जो plant breeding है, it is helped to develop several useful and important varieties of the certain crop plants such as tomato, tobacco, potato, and okara, etc. So this is about the breeding to obtain insect or the pest resistant varieties. Next is plant breeding for improved food quality. What are the various steps that are used against the plant breeding for improve the various food qualities? So plant breeding is important for improving the quality of the food as well as the meet the nutritional requirement of the food. So plant breeding it is also an important aspect for improving the quality of the food. एक तो quality of food को improve करने में एक important aspect है, as well as meet the nutritional requirements of the food. Diets, especially the plant-based diets की बात हम करें तो especially that diet based on the plant diets that lack essential micronutrients, जिसमें कि वो lack करता है essential micronutrients को, increase the chances of diseases. जो diseases के chances है उनको increase करेगा, reduce lifespan, automatically lifespan जो होगा वो reduce होगा, and reduce mental abilities, और automatically उस बिहार पर जो mental abilities होंगी वो भी क्या होंगी, reduce होंगी. The objective of plant breeding for improved food quality is to improve nutrient content. अगर जो main objective है plant breeding का, वो main objective क्या है plant breeding का? For improve the food quality, to improve the nutrient content, protein content, oil content, vitamin and the mineral content. So these are the main objectives of the plant breeding. Next is biofortification. What do you mean by biofortification? So biofortification is the process by which the nutritional quality of the food is enhanced. ये बायोफोर्टिफिकेशन जो है वो एक ऐसा प्रोसेस है जिसमें कि न्यूट्रिशनल क्वालिटी जो है फूड की दैट इज एनहांस्ड थ्रू एग्रोनॉमिक प्रैक्टिसेस, जो कि किसके द्वारा एनहांस होती है? थ्रू द एग्रोनॉमिक प्रैक्टिसेस, कन्वेंशनल प्लांट ब्रीडिंग and modern biotechnology is known as biofortification. So in 2000 maize hybrids were developed that had twice the amount of the amino acids. 2000 में जो maize hybrids थे उनको develop किया गया, and जब maize hybrids जो थे वो develop हुए उसका मतलब क्या हुआ कि they had twice, वो double हो गए किससे? The amount of the amino acids, lysine and tryptophan compared to existing maize hybrids. Next is the wheat variety having a high protein contents. जो wheat varieties थी इनमें high content जो था प्रोटीन का था. Atlas 66 has been used as a donor for improving cultivated wheat. It has been possible to develop an iron fortified rice variety containing over 5 times as such iron as in commonly consumed varieties. So all these are the consumed varieties. The Indian Agricultural Research Institute, जो Indian Agricultural Research Institute है न्यू दिल्ली में, has developed several vegetable crops. उसमें जो है develop किए उन्होंने several vegetable crops that are rich in vitamins. वो vegetable crops जो है they are totally rich in the vitamins and minerals etc. Vitamin enriched carrots, spinach, pumpkin, vitamin C, enriched bitter gourd, bathua, mustard, tomato, iron and calcium, encircle or enriched spinach and bathua and protein enriched beans, broad, lab, French and garden peas. So all these are the concepts that are related to the biofortification. Next is the single cell protein, and this single cell protein is very important according to the examination point of view, and there is automatically a question raised against this question, that is, what do you mean by single cell protein? It is an alternative source of the protein for both animals and the humans. ये एक alternative source है proteins का जो कि both helpful है in case of the plants as well as animals or the humans. Agible unicellular microbes can also be defined as, जो agible unicellular microbes हैं they also can be defined as a single cell protein. जो हमारी agible unicellular microbes हैं they can also be defined as a single cell protein. Next, spirulina is a rich source of protein. Spirulina जो है that is very rich source of the proteins, minerals, fats, carbohydrates and vitamins. This is also one of the method, ये एक important method है of reducing environmental pollution. Environmental pollution को reduce करने का जो है ये one of the most important method है. Next is tissue culture. What do you mean by tissue culture? Tissue culture is defined as the capability to develop the whole plant. Tissue culture को अगर हम define करते हैं तो that is the capability to develop the whole of the plant from the part of the plant, and this capacity to form whole organism plant is known as totipotency. और ये capacity जो develop करती है whole plant को या whole organism को, that capacity is known as the totipotency. For tissue culture, nutrient medium growth regulator, अगर हम tissue culture की बात करते हैं तो proper nutrient medium, proper growth regulators such as auxins, cytokinins are needed. तो अगर हम tissue culture की बात करें तो उसमें proper nutrient medium चाहिए, proper growth regulator चाहिए such as auxins as well as cytokinins are used. This method of producing large number of plants in a very short span of time. ये एक ऐसा method है जो कि large number of plants को produce करता है in a very short time is known as micropropagation. तो micropropagation एक ऐसा concept है जिसमें कि large number of plants को जो produce किया जाता है in a very short interval of time, and that concept, that method is known as micropropagation. It will produce genetically identical plants known as somoclones. तो ये जो है एक काफी जेनेटिकली आइडेंटिकल प्लांट्स हैं जिनको कि हम क्या कहते हैं सोमा क्लोन्स. तो मेरिस्टम्स सच एज सेल्स व्हिच हैव कैपेबिलिटी ऑफ रिपीटेड सेल डिवीजन. तो मेरिस्टम्स जो ये है वो एक ऐसी सेल्स हैं जिनके पास कैपेबिलिटी होती है किस चीज की? रिपीटेड सेल डिवीजन की. that are known as the meristematic cells can be used to grow virus resistant plants. वो grow करते हैं virus resistant plants को, such as banana, sugarcane, etc. So this is the tissue culture, and this is the last topic of this part number 2, that is the tissue culture.